tell us the transition to Raise, the story behind the transition to Raise.
Yeah. When I left Five Prime, I was not the founder of Five Prime. That was Rusty Williams.
Yeah.
So credit to him. I did come in when it was still private, but it was already in existence. But when I left Five Prime, I thought, I want to build a company, because I had to do some layoffs at Five Prime, and that was no fun. And I really, hated telling good people goodbye. I didn't want to take apart a company. That's not what was motivating me. I wanted to build a company. And so I wanted to do something from scratch. And so that was the idea for Raise Bio. And the scientific idea came from a former Five Prime colleague, Deb Cherish, who approached me about it. And immediately I started working with Deb kind of on a pro bono basis as I was leaving Five Prime and thinking about what I wanted to do next. Mm-hmm. First in a coffee shop, and then we'd get together wherever we could until the pandemic hit, which came about soon thereafter. But initially, it was just sort of giving her free advice. And then she asked me, and I decided I did want to be the CEO and put this company together with her and our third co-founder, Aaron Kantoff. So that was the triumvirate that formed what became Raise Bio. And the reason I did it is I'd been looking at the radiopharma space sort of as an observer, just being in the cancer area for many years and seeing things like a radium product that Bayer has now that got approved for prostate cancer, acquisitions by Novartis of a French company called AAAS and Endocyte that had targeted radiopharmaceutical products that are now approved as Pluvicto and Lutathera. And I saw that these things were quite active and that the toxicity could be managed. In contrast to older generation products from years ago, there were some radio-labeled monoclonal antibodies to CD in the early 2000s called Bexar. And there was another one, and they both had a lot of toxicity associated with them, mm-hmm. Bexar and Zevalin. But they were very active. So I thought if somebody could control that toxicity with that targeting, these would be very active agents. And so the idea that we had at Raise, and really Deb catalyzed this, was using a different targeting modality, which is peptidomimetics. So these are things that are macrocyclic. They form a circular type structure that resemble peptides. So you get that targeting and specificity that a peptide can give, but with a little bit better half-life. So they're not as permeable or not as likely to be damaged by proteases. So they get a little better half-life, but they're small enough that they can penetrate into the bed of the tumor. So we thought that would be kind of the Goldilocks targeting modality, because we wanted something that would stay in circulation for a little while, not accumulate in certain organs and damage them, but have enough time to do what it needs to do in the tumor, which is to locally irradiate those tumor cells and kill them. So we chose that as a targeting technology, which was a good move, I think. And then for the isotope, which is the warhead that emits the radiation locally, we went with a different isotope than has been used historically for commercial products, and that's actinium-225. So predominantly to that point, the isotope of choice had been lutetium, which is a beta particle emitter. So to get into the physics a little bit, a beta particle is basically an electron. So lutetium shoots off these electron-like particles. You got to have a lot of those hitting a cell to kill it. They're very small. They don't have the energy that an alpha particle does. So alpha particles are what actinium-225 emits. And you can think of these as like little cannonballs that come shooting off the atom. And one of those can damage the DNA of a tumor cell and kill it. So they pack a lot more punch. And we'd seen some data out of Germany and South Africa. Tumors that were not responding anymore to lutetium-based therapies were responding to actinium-225 therapies. And so we saw an opportunity there. That was the genesis for the sort of premise of what became Raise Bio, which was to combine these things, to marry these two technologies in a way that had not been done before, and to select very good targets to go after. So that was what we did at Raise. We struck deals to access the technology, both on the isotope side and the targeting side with the peptidomimetics, raised some financing. So that was all during the pandemic. It was all done like this over video conference. Didn't meet anybody in person then. This is sort of early 2020. It raised a Series A round and then actually built out the company in San Diego. And I left the management team at that point, stayed on as an advisor and investor, of course, but I wanted to do another startup. So that is what led me to Aulos, where I am today.
Yeah.
But, the general sort of decision criteria I've used all along the way is better targeting to the tumors or to the cells that can kill the tumors. So whether that was very specific small molecules or antibodies at Genentech, or again, a targeted antibody at 5' to FGFR2b, or targeted peptidomimetics with radiation attached at Raise Bio, or now at Aulos targeting a particular cytokine. It's all been about that, to try to limit the side effects, maximize efficacy, and to do so in a way that really changes the game for patient outcomes.
Yeah, very good. I'm looking at the clock and I'm like, wow, we haven't even really gotten to the Aulos story yet. So let's go there.
Sure.
Let's go there quickly. Yeah. So given your track record of picking winners based on those criteria, which you very, very clearly stated, what was it about the concept of Aulos, and how did the company come to be?
The company came to be through two co-founding entities. So ATP, the venture capital firm, had seen some technology from Biologic Design, which is an AI-based creator of antibodies. And in particular, ATP saw this program that was targeting the IL-2 cytokine, and proposed to Biologic Design the idea of creating a company around just that program. And that's actually what was the genesis of Aulos. So ATP had the vision and the foresight to see this program, realize that there was something there that with its own management team and financing might go faster, might go farther. And I think Biologic saw it the same way. And so the deal that set up Aulos was that ATP invested and contributed capital in the Series A round, and Biologic Design contributed the antibodies and the intellectual property and the know-how. And we continue to collaborate with Biologic Design. So they are working with us on a day-to-day basis with the ongoing research and development. They assigned the rights to the intellectual property, to all the antibodies to Aulos, and took an ownership stake in the company. So as the value of Aulos increases, their ownership value will increase. So it's been a very healthy, productive relationship. And kudos to both organizations, ATP and Biologic Design, for seeing the merit of doing this. And so I was hired as the first employee by ATP, and I came in as employee number one in early 2021, and then it built out the rest of the team here. But what it was that appealed to me was I knew about IL-2 from years of working in oncology, and I worked with clinicians who'd given IL-2, and they'd seen it melt tumors away. They'd also seen it put people in the ICU. So I knew it was a super active cytokine, very potent. Somebody just called it the OG of cytokines, or immuno-oncology drugs, and it is that. It's the first approved immuno-oncology agent dating back to 1992 in the United States, and it's still approved for melanoma and renal cell carcinoma. And when it works, it can work beautifully. You can get durable, complete responses, and they can last for years, like 5 to 7 years median for these durable, complete responses when they occur. Now, not every patient experiences that, and many patients experience a lot of toxicity when they receive high doses of IL-2, because in addition to activating the immune system, there are receptors for IL-2 that line the vasculature on the endothelial cells and on the smooth muscles around the vasculature that change the diameter of those vessels. They also express that same target. And so there's an on-target effect of IL-2 in that it causes vascular leakage, which means fluid comes out of the blood vessels, the blood pressure drops, the patient swells up, the heart rate often goes up, and this can be lethal. So lots of room for improvement in therapeutic index, but I knew it was a very active cytokine. So when I heard about what Aulos was contemplating this antibody, a human monoclonal antibody that would bind to IL-2 and keep it away from immunosuppressive T regulatory cells and the vasculature, but allow it to bind to the effector cells that can kill the tumor, like CD8 T effector cells and NK cells. I thought that sounds like a really good idea, but I'm not the immunology expert. So I reached out to some friends and former colleagues who did know immunology and just doing my diligence on it. And they said, yeah, that's a really good idea. If it works, that could be a game changer. Mm-hmm. So that gave me the motivation that I needed to accept the offer. And I'm very happy I did. It's been a great experience, and I've built out a wonderful team, and I still get to work with the people at ATP and Biologic who put this company together, and a lot of brilliant minds around the table.